Hello, everyone. This is Dr. Asi. Welcome back to the channel, Dr. Asi Explains. Today's topic is BSC1011 Lab, Module 1. Module 1 includes 5 labs, lab number 1, 2, 3, 4, and 5. So lab 1 and 2 are about systematics and also including phylogeny, which is taxonomy plus systematics. Taxonomy plus phylogeny, which is systematics. Lab 3 and 4 are Protista, the kingdom, and lab 5, starting with Plante, like Bryophytes. So let's go ahead and start. We are going to review each of these labs 1 to 5. Okay, so let's get started. Okay, so lab 1 is just basic systematics. So as you know, systematics is a combination of taxonomy and phylogeny. Taxonomy is about classification of organisms. Phylogeny shows evolutionary relationship between organisms. So that's really explaining adaptation of organisms, unity, diversity of life, includes fossil records, which basically remains of traces of past organisms that they disappear, but we actually know them from their fossil records. If environment changes or individuals or new environments, adaptation to the new environments may give rise to the new species and populations and basically changes and evolution of organisms like bacteria, virus, other stuff. They are all linked to each other in this systematics. Now, there are keywords like homology, which is basically if there are two things they are homologous, their similarity is due to common ancestry. So, like example, genetic code, which is all organisms use same genetic code. Genetic variation makes evolution possible. And we have a game in this chapter, we have a big clock with 4.6 billion years with different parts in it and which each organisms came out. So make sure you study that, and also you look at these cladograms like tree of lives in taxons in each unit, and then branch points that actually they share between the branches, sister taxa and basal taxon that we cover in lab 1 and lab 2. So that's lab 1 and 2. Lab 3, 4 are about Protista, which is basically the one of the kingdoms that we cover in 1011. Protists are a eukaryotes, but mostly unicellular eukaryotes. So we see five supergroups under protists, including excavata, chromalvolata, rhizaria, archaeoplastida, and uniconta, right? 5. So they all look like different things. Like uniconta look like fungi and animals. Archaeoplastida, they look like plants. Rhizaria, it's like we only know them from fossils mostly. It's and some definitions like DNA similarities. Chromalvolata, like algal, like diatom, brown algae. So those groups, and excavata, basically they have we know them because some important species under excavata that they cause actually some important diseases. So in animals and humans. So make sure that you actually study this lab 3 and lab 4. So each of these super groups that we cover basically under Protista, right? So like Euglena, and how many flagella they have. Flagella is this long, tall one, and cilia is this short, like hair-like structure. And they might have one flagella, like Euglena, or two flagellas, like Chlamydomonas. what disease what is causing what disease, like sleep sickness and some other diseases, or algal blooms, which one of these organisms causing algal blooms, which one causing malaria, right? So pay attention to each of these supergroups, and then their structure under microscope. So there is one slide of the very last slide in each slide group that actually in my slides that summarizes all of those that you need to know that they are most important in each lab. That's lab 3. Lab 4 continues with Archaeoplastida, unicorns, and then some examples of these. It's just continuation of protists. Okay. Lab 5, starting with plantae, even though lab 5 we don't finish plantae because we are going to actually continue in coming up labs with the plantae, but this is the very first group in the plantae, which is Bryophytes, non-vascular plants, and has three phylums under: liverworts, mosses, and hornworts. Even though in the lab it doesn't cover hornworts, it's mostly focused on liverworts like Marchantia, mosses, basically the 2 phylum, but not the phylum number 3. So pay attention. We're starting here. We start seeing actually life cycles, which is made of gametophyte and sporophyte, and 2 processes, right? So meiosis and fertilization. So it's alternate. So it has a name called alternation of generation. And also pay attention, Antheridia, Archegonia, which is male or female, right? And so what does each of them look like in each phylum? And also there is asexual reproduction, which like gemmacops, which can actually without fertilization, they can actually go and make a new bryophyte. That's possible in plants, and those are important too. So just 2 phylums you need to study under lab 5, and overall basically lab 1 to 5. So everything we covered in the lab, study your notes, look at the PowerPoints, look at all the slides and specimen in the lab. They are lab 1 to 5. I mean 3, 4, 5 have the specimen and my slides, microscope slides. Lab 1, 2 is just intro and phylogeny. There is no specimen. Basically, it's all about life cycles and the tree of life and cladograms. So thanks for watching, and we will do a review actually in next class. And if you have more questions, just basically send an inbox through Canvas. Thank you for watching again. Don't forget to subscribe, like, and share the video, and see you in the next video.